different because I like to move about, and I'm afraid if I fall off the podium, I might have a TBI, so I better keep my feet on the ground. In the first slide, let's see how that works. Can I do this? Okay. Actually, no, there's one here. Yes, for anyone who has any knowledge of American baseball, you might recognize this player, Lou Gehrig. He played with the New York Yankees in the 1920s and 30s. Very famous. He played also with a player called Babe Ruth. Anyway, the story is that in 1939, he retired from the game voluntarily for health reasons and died several years later. And this made very big headlines in newspapers. He died of this rare disease, which was named after him because of Lou Gehrig's disease, or ALS, motor neuron disease. Actually, ALS is a motor neuron disease. It's not the only one, but probably the most prominent one. Now, ALS is, most cases are sporadic. A small percentage are hereditary. And of these which are hereditary, a small number have a mutation in the enzyme superoxide dismutase. Now, unlike most mutant enzymes, this one is important for cells' antioxidant defense. Unlike most enzyme mutations, this one is not a loss of function, it's a gain of function. How the gain of function causes disease is still not fully understood. Actually, this mutation, there are several in this enzyme, form a very small percentage of all ALS cases. But it has been used to create transgenic models of the disease, which mimic in many respects the human form of the disease. Now, what happens in ALS is that you lose upper and lower motor neurons in the motor cortex, the brainstem, and the spinal cord. And essentially, these innervate muscles in the legs and the arms. You have difficulties with ambulation, difficulty grasping objects, difficulty breathing, and most patients die after 3 to 5 years of diagnosis of respiratory failure. Now, what causes this is still not known. Their motor neurons develop these protein-rich inclusions, which contain ubiquitin and one or more proteins involved with ALS. So it's still a very complicated disease and not fully yet understood. Now, the disease, when they first made mouse models, they expressed the mutant protein in all the cells, in neurons, in glia. And then someone came along and they made mouse models in which they expressed the mutant protein in neurons only. They did not get disease. So if you express the mutant enzyme in glia, you get the disease. So glia are important for expression of the disease, at least in these models in mice. Now, the disease is accompanied by an inflammatory component. Glia are important, of course, in the nervous system. They have a number of neurons. Microglia are important. They phagocytose debris. Astrocytes form part of the blood-brain barrier. They take up glutamate from the synaptic cleft, make trophic factors. Lymphocytes produce myelin. But these cells, the microglia and astrocytes, if they become overactivated, produce inflammatory agents which can damage neurons. Now, normally you have a crosstalk between neurons and glia. In ALS, with its inflammatory component, you have a disconnect in this crosstalk. So both microglia and astrocytes develop a pro-inflammatory phenotype that produce agents which can damage neurons. Those lymphocytes are also involved not because they're pro-inflammatory, but because their health can be compromised. They produce energy for neurons, which if that is reduced can damage neurons. They also will produce less myelin, which can affect axon transport. So, okay, given that there is this inflammatory component, there's also the role of the immune system, besides microglia, which are part of the immune system. Lymphocytes have a role, but then we need also for other types of immune cells. One, which we're interested in, are called mast cells. Now, mast cells probably are not as well known as microglia or lymphocytes. Mast cells derive from precursors in the bone marrow. They are part of the innate immune system. They have some similarities to eosinophils. They produce migraine in the blood, but like eosinophils, they remain immature until they reach their tissue location where they sit down, immature, and become mast cells. Mast cells are heterogeneous. Their characteristic depends upon the tissue in which they are expressed and also the animal species. Now, mast cells go back a long time. They were first described by Paul Ehrlich in 1878 because they have these rather peculiar granules in the cytoplasm. These are stained for histamine. When they become activated, they degranulate. This one is in the brain parenchyma. These granules come out, they can travel quite some distance, about 50 micrometers, they cover quite a bit of territory. So mast cells tend to be located at sites in the body close to the environment. They're in the skin. When you have an insect bite, the swelling is histamine released from mast cells. They're in the bronchial airways. If you have allergy to pollen, they release histamine, which causes an asthmatic reaction. They are perivascular in location. They're in the brain meninges. You find them close to blood vessels, to nerves, around here. So you can imagine if, when they degranulate, this rather large footprint will cover quite a bit of area. They are involved in hypoxic ischemic brain injury. They are seen in MS around plaques. They are involved in breakdown of the blood-brain barrier, and one may ask, are they involved also in ALS? Now, looking through the literature, possible data, there are some suggestions that, in fact, it might be the case. Mast cells do release a lot of different things in the granules, histamine, proteases, which can be tissue degradation, heparin, serotonin, cytokines. A number of these are also made de novo. Again, cytokines, growth factors, free radicals. They express T B cell ligands, so they can also participate in cell-to-cell contact with immune cells like lymphocytes. So evidence, well, we look through the literature and some things they do. If you look in ELS tissue, you will see IL-17, the pro-inflammatory cytokine, expressed in the spinal cord. This is grade 9 for PLS. These cells are stained for IL-17. Now, if you look more closely, you see cells stained for IL-17, which also express CD3 lymphocytes. These cells express tryptase, which is mast cell selective enzyme. So you find both IL-17 expressing T cells and mast cells in the gray matter of patients with ALS. These patients also will have increased levels of IL-17 in the circulation. It tends to be higher early on in the disease. In spite of this scatter, it appears to be significant. The controls are shown over here. The scale is logged, so from 1 to 2 and 2 to 3 is a tenfold difference. So there's quite a big difference between the patient serum and the controls. Again, mast cells and T cells can interact. Mast cells release IL-6, which can induce IL-17 production by T cells. Now, T cells and mast cells are these agents which can attract each other. One, T cell agents will activate mast cells, and antigen presentation by mast cells will activate T cells. What you can get is what some people have called an immunological synapse, in that mast cells, by presenting antigen, will actually cause the secretory mechanism of T cells to become polarized towards the mast cell, and you form what is essentially a synapse, in a certain sense, between a mast cell and a T cell. So the two do interact with each other and can accentuate a pro-inflammatory response. IL-15 and IL-12 are increased in sera and CSF of ALS patients. In this study, we looked at, this is the sera, this is the CSF for both IL-12 and IL-15. And there was a difference, significantly increased for both of these cytokines. Now, mast cells produce IL-12, and IL-15 is a chemoattractant for mast cells. You can show this using what is called a Boyden chamber. So if you put your cells in the upper chamber and your agent, this is leukotriene reform, but if you put IL-15 down here, the cells will migrate across the chamber. So if you put IL-15 in the lower chamber, mast cells in the upper chamber, they will migrate, and it is concentration dependent. If you put IL-15 in both the top and bottom, you essentially neutralize the gradient and lose the migration. So again, it's possible that by producing IL-15, these cells may induce migration of mast cells into the spinal cord, just as they migrate across the blood-brain barrier when the barrier is compromised in, for example, ischemia. An interesting feature of ALS is that these patients often experience neuropathic pain. Mast cells appear to have an important role in pain transmission. If you look at the pain transmission pathways, pain or nociception will start in the periphery, but it's integrated and expressed in the CNS. So if you look at peripheral tissue, which contains mast cells, an noxious stimulus causes release of mast cell mediators, which can lead to sensitization of primary nociceptors, first order neurons, which transmit the signal to the spinal cord, the dorsal horn. And in the dura mater of the spinal cord, you find mast cells as well. Again, they become activated and can sensitize neurons in the spinal cord, second order neurons, which then pass their signal through this gateway, the brainstem, to the thalamus. The thalamus is very rich in mast cells. When you have a situation of pain, they become activated. They release agents which can activate neurons in the cortex. In the end, you have expression of pain. If you take and injure a sciatic nerve and look in the thalamus, you will find several hours later increased numbers of mast cells and activated mast cells in the cortex. So mast cells very likely participate in pain transmission both spinally and supraspinally. So potentially they participate also in the pain which these patients experience. Curiously, animals which are engineered to overexpress this mutant enzyme SOD and which have features of ALS, often will also show features of neuropathic pain. So the message to all of this, 2 points, is that I think when you, if you look at inflammation in ALS, one should consider not only the role of microglia and astrocytes, but perhaps also mast cells. If you were to take and design an anti-inflammatory strategy for ALS, you should probably think about targeting the microglia astrocyte mast cell triad. So the last slide is sort of a cartoon in which I've indicated that astrocytes, microglia, and mast cells do interact with each other and together can impact aggressively and inversely on the central neuron. Now, evidence for their interaction, there's quite a bit which is a talk in itself. Mast cells and microglia interact. For example, mast cell, an enzyme called protease activated receptor 2, which is cleaved by activation of mast cells through microglia cytokines, will activate microglia and release inflammatory agents. These two also work through the complement system. Microglia express complement C5a and its receptor, which are upregulated when they become activated. Mast cells express C5a receptor, but not C5a peptide. However, C5a is a very strong phenotactic for mast cells, which again can lead to these two cells interacting. So I think the bottom line is that when you look at inflammation in the CNS, one needs to consider all three cell populations. So in the interest of time, I think I will stop here and take any questions.
Thank you very much, Professor, for this very interesting talk, fascinating. Any urgent questions? Otherwise, yeah, please.
No,
Yeah. Microphone will come.
Thanks the compliments. Of course, the inflammatory theory, is fascinating in all the field of neurodegenerative disorders. I would like to make a point as a clinician. I have seen hundreds and hundreds of ALS in my career, but I never found pain. Sensitive symptoms, by definition, are not present in ALS, sensitive symptoms. Of course, you can get pain for instance, arthrosis or other things, but not neuropathic pain. That's to make a point of, of this field. That's it.
Yeah, mast cells, apart from, neuropathic pain is one of the places where mast cells are involved also in inflammatory pain as well, besides neuropathic pain.
Oh.
They might be.
All right, thanks a lot. There will surely be a chance to discuss this later in the coffee session, coffee break after this. And I hand over the chair of the session to Peter.
So the next speaker.